Kendra. It's really a pleasure to be here today to talk to you about a topic that is very near and dear to my heart. And again, thank you for mentioning the two podcasts that I do. The URLs for those are shown here on the title slide. It's foodsafetytalk.com and also riskyornot.co. If you go to riskyornot.com, you'll find some guy giving you investment advice, which is not me. So please do go to riskyornot.co. All right, let's get into it. So I've entitled this presentation, A Food Microbiologist Looks at Hand Washing and Hand Sanitizers, because I came to this field from the perspective of a food microbiologist, but there's a lot of people that do research in hand washing and hand sanitizers that are, for example, come from the medical world, where I think many of you may come from as well. And so I certainly have a unique perspective as somebody who thinks about food and cross-contamination and hand washing in the context of food preparation. But let's get into it. So this is, for me, where it all started. And this is a paper by Yuan Chen and colleagues. And this was really our first foray into trying to understand hand washing as well as cross-contamination in a kitchen environment. And although this paper was published roughly 1/3 of the way into my career, it has far eclipsed our other publications in terms of citations. It really started, I don't know if revolution is the right word, but it really did get people's attention, and the work that we've done in this area has really influenced the work of many people that have come after us. But like so many things, if I had it to do over again, I would probably do it a little bit differently. And this is the figure that I should have included, because I think there's still a lot of people that get this particular point wrong. And again, and realize too that the perspective that I bring to hand washing and hand sanitizers and things like that is a quantitative perspective. And so if you look at the work that my lab does generally, it is really focused on counting microorganisms, right? And that's why my Twitter handle is bug counter, right? Because microbiologists refer to microorganisms as bugs. It has nothing to do with entomology or insects. And so we do like to count things in my lab. And I still remember to this day when Dr. Chen brought some data in from when we were first starting to get some data from this project, and it looked exactly like the curve on the left, where if we plotted the percent transfer of microorganisms versus the frequency, the number of participants in the trial that showed a certain percent transfer, we saw a graph that looked very much like the one on the left. And I knew enough from all of the work that I'd done in my career during the 10 years previous, looking at statistics and transformation of data. I knew that those data, and you probably know this too if you ever had a class in statistics, the data on the left are not normally distributed. And one of the first thing they teach you in a traditional statistics class is all of the statistics that we are going to cover in this course all assume that the data are normally distributed. And so those data on the left are not normally distributed. But it turns out you can apply what's called a transformation to the data. In this case, we simply take the logarithm of the percent transfer of microorganisms, and all of a sudden you see a curve that is much more normally distributed. Now, it's still got some noise in it. It's still not perfect. If we collected more and more data, we would see increasingly, better and better accuracy there in terms of transformation of the data, and it would gradually, I think, approach a normal distribution. And so, again, this was one of the things that I think we contributed to the literature that was really important, is you have to analyze the data correctly. And the other thing too that was somewhat unique about this work is we didn't do 3 experimental trials, right? Often when you're doing science, you do it 3 times, and if you get 3 answers that are relatively reproducible, you're done. Well, for this, we really knew there was a lot of variability from person to person. So we did experiments where we had 30 participants. And if you apply an incorrect statistical transformation and you only study 3 people, well, you're going to get data that don't necessarily make sense, or at least are not accurate or representative representative of the situation at hand. And so, and anyway, that's probably the most nerdy and math and statistically oriented I'm going to get. But I do think it's an important point that I try to communicate, because I think even after we published our papers, people still get this wrong. Okay, so flash forward now, that paper that I just talked about was published in 2001, flash forward to 2007. And the other thing too that's interesting about this work is that we do other research as well. And so we'll dip into this area a couple times. It's not like we're continuously working in this area. We'll work on it for a few years, we'll go off and do some other things, we'll come back when we get some more funding, and we'll keep on going. And so this was some really interesting work that we did with a large food service chain. You would recognize them. They sell lots and lots of hamburgers. They even have a sign outside the restaurant saying how many hamburgers they sell. And they were interested in figuring out what would happen if the workers in their restaurant forgot to wash their hands or didn't wash their hands after handling frozen hamburgers, and then went on to either use a hand sanitizer or to do nothing at all, and then begin to make a salad where they're touching lettuce leaves with their contaminated hands. And so basically what we did was, this is another interesting aspect of the work that we do in my lab. It's really a combination of math and statistics and computer models, as well as actual laboratory data. And so in this research, we did some actual data collection in the laboratory, but then we also used math and statistics and some calculations and data from other papers. And so basically we calculate what's the chance of somebody getting sick from eating a salad from this restaurant if somebody didn't wash their hands. And it turns out that risk is actually relatively low. And so we didn't actually calculate the probability of illness, but we did calculate the probability that a single cell of a pathogenic organism, E. coli 0157H7, that's found on the outside, that's found in ground beef, we calculated the probability of, if somebody failed to wash their hands, what would the chance of them actually getting an organism, an E. coli organism, on a piece of lettuce? And the answer is actually, it's surprisingly low. It's on the order of about 1 in a million. And so if you have people handling burgers and you have them making salads, you would never detect a single E. coli until you did that about 1 million times, which said that the risk was actually relatively low. And part of the reason why the risk is relatively low is that ground beef typically does not have E. coli in it, pathogenic E. coli. And then if it does have it, it's probably not on the surface of the patty. And if it is on the surface of the patty, chances are it doesn't transfer to somebody's hand. But if it does transfer to somebody's hand, chances are it doesn't actually transfer to the lettuce leaf. So the risk is not zero, but it's very, very close to zero. And also, the most important thing we can do in that case is to properly cook the hamburgers. So there's really little, in this case, little risk of cross-contamination. Okay, so having gotten involved in hand washing and having done research in hand washing, I was approached by a company that is a trade association for companies that sell soaps and detergents. They used to be called the Soap and Detergent Association. Now they're called the American Cleaning Institute. And they said, well, you know, we're hearing rumblings about the potential banning of antimicrobial soaps. And one of the reasons why people want to ban antimicrobial soaps is that they don't actually work. They don't actually work to kill microorganisms. And they said, would you be interested in taking a look at this? Would you analyze data from the scientific literature? And so I collaborated with my colleague, Rebecca Montville, and we published this paper. And I'll give you kind of the key take-home message here. So what you see here, it's another frequency plot, and we do like to show frequency plots in the work that we do, because very often we're trying to get a handle on the variability of data. And what you can see, and this is data that we collected from the scientific literature, where people studied antimicrobial soaps and non-antimicrobial soaps, and what was the effectiveness of that soap at reducing the bacterial population on the hands. And so we measured the bacterial reduction in terms of the logarithm of the number of colony forming units. So it's basically the logarithm of the count of the number of microorganisms on hands. And what you can see here is the non-antimicrobial soap, that's shown by the black symbol, and the antimicrobial soap, that's shown by the gray symbols, the gray bars. And what you can see is that those curves do kind of overlap with each other, right? But if you do a statistical analysis, what you'll find is that although the curves overlap, the antimicrobial soaps are actually slightly more effective. They're about a half an order of magnitude more effective, which doesn't look like much, but actually when it comes to risk, that half a log reduction can make a big difference. And of course, and we're going to come back to this, because what we did after having done this work, we actually came back and did some follow-up work based on some laboratory data. So what we did here was analyze the scientific literature, but in the subsequent work, we use actual data collected in a testing laboratory, and we further investigated. And again, we'll come to that in a little bit. Okay, so one other really interesting discovery that we made as part of this same work is that it really mattered what it was that you were studying. And so you can kind of divide the scientific literature on hand washing into three different types of experiments, okay? The first type of experiment is where you deliberately put gram-negative bacteria on people's hands. And so these are bacteria that stain negative with a gram stain. And so this includes organisms like Salmonella, E. coli, just to name a couple. You can also do experiments where you deliberately put gram-positive microorganisms on people's hands. And so these might be experiments with organisms like Staphylococcus aureus or Listeria monocytogenes. But there's this third type of work that people do as well, and that's to use the naturally present resident flora on people's hands. And so again, one type of research, we're going into the laboratory, we're deliberately adding high levels of microorganisms to people's hands. And in the other, we're just basically taking whatever they give us, looking at the level. People have two hands, right? And so you can invite somebody into the laboratory. You can test one of their hands for bacteria. You can have them wash their hands and then test the other hand to see the difference. Sort of assuming that you have roughly the same concentration of bacteria on your two hands at one time. And what we see here is that studies that were done with the resident flora, the naturally occurring bacteria on people's hands, hand washing is much less effective. And I won't get too much into detail here, but I think that the reason why is, and this is something that we've seen repeatedly and in the literature and also in our own work, is that basically the more bacteria that you put on people's hands, the easier it is to demonstrate the effectiveness of hand washing. So the more that you put on, the more that you can measure, the more that you can take off. But if you start with a really low level, like most people naturally have on their hands, it's a lot harder to show the effectiveness of removing bacteria. So, and again, we'll come back to that. We'll touch on that theme again at some point, or if not, we can talk about it in the Q&A. So all of this work that's been going on at this point in my lab for almost 15 years, all of this was really done on a shoestring budget. The initial work done by Yuan Chen and colleagues, we got a tiny little grant from the Sloan Valve Corporation that was interested in quantifying the effectiveness of touch-free faucets. We also got a similar small grant from the hamburger fast food company. Again, not full grant funding, but just a little shoestring money to do a small project and publish a paper. This was part of a grant that we got funded by the Food and Drug Administration. And so in this work, we quantified the effectiveness of a variety of factors. How long do people wash their hands? Do they use soap or not? Is there debris on their hands? Do you have ground beef present on your hands or not? And then also look at the effectiveness of drying methods. And again, there's a lot to detail in this paper. I probably could spend the whole time talking about it, but I won't. But one thing that I really do like, and this was work done with my colleagues, Michelle Daniluk from University of Florida and Linda Harris from UC Davis, but it was work that was actually done by my graduate student, Dane Jensen. And one of the things that Dane really wanted to look at was something that he calls the splash and dash. The splash and dash, as defined in our research here, is a 5-second hand wash with no soap, right? A lot of the published research on hand washing uses a quote unquote proper hand wash. And so we wanted to compare what's the effectiveness of a proper hand wash to the improper, but we know often used splash and dash. And so what we see here is that based on the methods used in this paper, the splash and dash actually gives you about a 1 log reduction of the concentration of microorganisms on your hands. And a 1 log reduction, for those of you who are not used to thinking in logarithms, is about a 90% reduction. And so, removing 90% of the bacteria on your hands is pretty good. It's certainly better than nothing. But how effective is that compared to the proper 20-second hand wash where you use soap and you scrub your hands properly and all of that? Well, the mean log reduction for a proper hand wash is only about 2 logarithms. And so what that means is that has an effectiveness of removing about 99% of the bacteria on your hands. Now, if you have 100 bacteria on your hands, and you remove 99%, that's great because now you only have 1 organism on your hands. But if you have 100 million bacteria on your hands and you remove 99%, you've still got 1 million bacteria on your hands. And so hand washing as an intervention, at least as food microbiologists think about it, is not really very good. In the food microbiology world, we're commonly looking at, when we're cooking a food, we want to see a 5 log reduction or a 6 log reduction. So a 1,000,000-fold reduction, or 99.9999% reduction. So is hand washing good? Yes. Is proper hand washing better than the splash and dash? Yes, but the splash and dash in and of itself is, again, better than nothing. Now, another interesting finding from this work was looking at paper towels. So what we did was we had people wash their hands, and then we had them either let their hands air dry or dry their hands with a paper towel. So in this series of experiments, we were quantifying the effectiveness of just the paper towel at removing bacteria from hands that had already been washed. And so what we found was that if you used a paper towel, that actually got more bacteria off than if you just let your hands dry in the air. Now, I want to mention, we did not study in this work, and nor have we ever studied actually those really annoying hot air hand dryers, or the less annoying but still annoying Dyson Airblade type products. And so obviously in those products, if you're physically removing water, the water may contain microorganisms. But where are you removing them to? Well, you're removing them probably to the bathroom environment or the hand washing environment. So what we showed in this work is that yes, you actually do get a benefit from drying your hands with paper towels. Why is that? Well, those paper towels are physically removing moisture and hence bacteria that were remaining on your hands after the conclusion of the hand wash. So are paper towels a good thing? Well, from the point of view of removing bacteria, yes, they certainly are good. But also what that means is that any bacteria that you have removed from your hands with the paper towels are now actually going to be on the paper towels. And so now you've created another potential source of infection, which would be the paper towels that now have these microorganisms on them. So, I mean, what's the solution here? Well, I mean, I think there's advantages and disadvantages of both air drying and paper towel drying. And of course, there's environmental consequences to use of paper towels. There's environmental consequences to the electricity costs from running a hot air hand dryer, et cetera, et cetera. And you know, it's complicated. But the bottom line, the take home message here is that paper towels do work to remove more bacteria than just the hand wash, but now you've got to deal with the bacteria on the paper towels. Okay, so we're gonna jump back, and we skip forward another year or so, and we're gonna jump back now and talk about some more work that we did with antibacterial soaps. And so here, this is a collaboration with a bunch of scientists that work for soap and detergent companies, as well as the American Cleaning Institute, as well as a testing lab that tests microorganisms on people's hands. And so what we did in this work was we looked at laboratory data on looking at different products on removal of bacteria from people's hands, Shigella bacteria in particular. And then we said, okay, what if somebody washes their hands or treats their hands in these different ways and then handles a food product, and then people eat that food product, do they get sick or not? Now, we didn't actually feed food to people, okay? We used computer models to predict the probability of illness from the ingestion of microorganisms. And we'll get to that in just a minute. But first of all, let's look at the 5 basic treatments that we used. So we used 2 non-antibacterial soaps, and those are shown in the first 2 columns, okay? And these are, I forget exactly, I forget what the first one is. The second one is a product that's abbreviated KMF, which I think stands for Kiss My Face. It's like a very gentle baby soap. The third bar is antibacterial soap that contain the agent chlorhexidine. The next one is antibacterial soap with the agent triclosan. And then the third one is not a soap at all, but it's just simply ethyl alcohol. So using it like an ethanol-based hand sanitizer. And what you can see here from the error bars and from the letters is that clearly antibacterial soaps with chlorhexidine and triclosan, or ethanol for that matter, ethyl alcohol, all were more effective than the 2 non-antibacterial soap products. And in fact, also ethyl alcohol was better than chlorhexidine, but triclosan was sort of in the middle of those 2 and not significantly different from either one. So now what happens if we assume that this is the effectiveness of our hand wash, and now we do a computer simulation where we assume that the worker, prior to washing their hands, has 1 million Shigella bacteria on their hands, and then let's assume that we feed these, so the worker has Shigella on their hands, they do one of these 5 treatments to remove or kill the bacteria on their hands, and then they handle melon balls with their bare hands, and then the melon balls get eaten by a consumer. And this, again, this is not taking place in the real world. This is all inside a computer simulation, but it's based on data from the real world. Oh, and then, yeah, so T stands for tone. So, and what you can see here, the 2 non-antibacterial soaps are shown on the top, and the 3 antibacterial treatments are shown on the bottom. And so the y-axis is again frequency, so yet another frequency plot. And then the x-axis is the simulated number of cases out of 100. So we have 100 people eating these melon balls, and then a certain percentage of the time, different number of people get sick. And so what you can see here for the 2 antibacterial products, most commonly, you have about a 50% chance of illness. So about 50% of the people that eat the melon balls get sick based on, you know, the effectiveness of the hand wash, based on the assumed dose response model for Shigella, okay? But for the antibacterial products now, again, and here you can see, because of the shapes of these curves, they're somewhat skewed distribution. So it's not really meaningful necessarily to talk about the mean number of illnesses, right? But we could talk about the most common number of illnesses. And what you can see here is that the most common number of illnesses when we're using these products is probably about 5 people getting sick. Now, in some cases, you do get 50% of the people getting sick, or in some very small number of cases, 60% of the people. But most of the time, most of the people eating these simulated melon balls, these simulated people eating simulated melon balls, most of the time, most of those people don't get sick. And so what this is showing, and we also show this for different starting concentrations of microorganisms. And again, I'll also, it's not shown on this graph, but it is shown elsewhere in the paper, that in fact, these products, these antibacterial products, really are effective at reducing illness. Okay, now we're going to change gears again, and this is some more work done by my graduate student, Dane Jensen, and the title of this slide is "Hand Washing is Confusing." And this was some work that, again, Dane was a wonderful graduate student, very entrepreneurial, and this is an idea for a project that he came up with entirely on his own, which is some work that we've entitled Quantitative Analysis of Recommendations Made in Hand Washing Signs. And so what Dane did was he went out on the internet and he found examples of hand washing signs that are used in different companies or in different states, and we did a quantitative analysis. And so what you can see here, he found 81 different and unique hand washing signs, and you can see that there really are quite different. And so, for example, some of the hand washing signs indicated lathering, some indicated rinsing, some indicated an overall time that you should wash your hand. And then there's some signs that just told you to wash your hands. They didn't tell you how long to wash your hands. And so what you can see is that almost 30% of the hand washing signs we found didn't tell people how long to wash their hands. About 45% specified the length of time for lathering. A very small percentage talked about the length of time for rinsing the soap off your hands. And then about 71% of the signs indicated an overall amount of time to wash your hands. But you can see that the time differences between the different signs were also different. An average lather time of 18 seconds, a rinse time of 13 seconds, and an overall time of about 22 seconds, with the median, min, and max as you see there. And even was one sign that recommended that people wash their hands for 60 seconds. Now, in the FDA model food code, it's a 20-second hand wash. But obviously in healthcare situations, and certainly if you're doing a surgical scrub, the amount of time may be longer, and this may reflect that. So clearly, if people are relying on the signs that they see to tell them how long to wash their hands, they're probably going to be confused because they're seeing different information all the time. Okay, I want to change gears once again and talk about a little bit of the work that we've done with bulk soap. Now, what is bulk soap? Bulk soap is soap that is placed in a dispenser where that dispenser is refillable, okay? And so, and this kind of soap that is dispensed in this way has actually caused outbreaks, not associated with food, but certainly there have been examples of contaminated bulk soap that have caused outbreaks of organisms spread from the soap in the hospital environment. And so, and this is work that was funded by a company called Gojo, which is a company that makes the Purell hand sanitizer, but they also make soaps, and they're very interested in public health, and they've gotten very interested in this idea of bulk soap over the years, and they've published a bunch of papers, including this one where we collaborated. And so what we found in this bulk soap study, we collected 296 samples of bulk soap. We did it in 3 different states, in Arizona and New Jersey and Ohio. And what we found was that 85% of the soap samples didn't contain any detectable microorganisms. And that's a good thing. But when we did find contaminated soap samples, which again is 15% of soap samples, 15% of bulk soap samples, it was most likely contaminated at a very high level. 10 million bacteria per milliliter of soap. And in this work, we also found a bunch of different types of microorganisms. We found Klebsiella, we found Serratia, we did find Shigella, the foodborne pathogen Shigella, in one sample. We found Enterobacter, we found another Serratia, and we found another Enterobacter. And so lots of organisms can happen. And we think what's going on in these bulk soap contamination examples is that and we mostly sampled food service establishments here. So we're looking at restaurants, we're looking at convenience stores, we're looking at supermarkets and things like that. And again, depending upon what restaurant or supermarket or convenience store you walk into, you may or may not see refillable soap dispensers. There are many companies out there that we just didn't go into their operations because we knew that they don't allow refillable soap containers. It's a single-use soap product that is, when it's done, you pull the whole bag out and you put a fresh bag in. And maybe that's less ecologically friendly, but it's way better in terms of potential safety. So what we think is going on in these open refillable systems is that very often, if you have one of these systems, you're maybe looking to save a little bit of money, and so instead of using full strength soap, you'll dilute that soap by 25% or 50%, and that will get you more mileage for your dollar. But it also basically begins to break down the preservative systems in these soaps. And lo and behold, given enough dilution over a long enough period of time and maybe some contamination coming in from the outside environment, these soap dispensers will become colonized with bacteria. And then once the soap dispenser is colonized, it very quickly gets up to this 10 million bacteria per milliliter of soap. And then once that dispenser is colonized, you just keep putting in fresh soap and you just keep maintaining those levels of bacteria. And so we think that open refillable bulk soap dispensers are a bad idea. Like I said, they're already banned in hospitals. They have not yet been banned in restaurants and convenience stores and supermarkets, but we would really like to see that happen. And so we keep trying to convince the regulators this is something they need to do, and we'll keep trying probably forever, or until they cave in and they finally ban these open refillable dispensers. We also did some work on the effectiveness of water temperature for hand washing, and so this is some work that was published in 2017, also work that was led by my former graduate student, Dane Jensen. And basically what we did was we studied 3 different water temperatures. We studied 100 degrees Fahrenheit, we studied 80 degrees Fahrenheit, and we studied 60 degrees Fahrenheit. But because this is published in the scientific literature, that corresponds to 38, 26, and 15 degrees C. And what is shown in this graph is not the hand washing effectiveness, because that's a much less exciting graph. This is the energy consumption for heating the water for 1,000 hand washes. And you can see that in fact it does lead to some significant energy costs to heat water to 100 degrees Fahrenheit for hand wash. What did we find in terms of the effectiveness of hand washing? Well, water temperature really did matter. There is a lot of variability from person to person in terms of how we wash our hands and the effectiveness of the washing of our hands, but water temperature is not a key variable. You can have an effective hand wash in 60-degree water, in 80-degree water, or in 100-degree water. You probably don't want to get too much warmer than 100 because it's not too comfortable, and even 60 degrees might be kind of cold for some people. And so what we ended up recommending from this work is that really what we want is water temperature that is comfortable. Why? Because if the water temperature is comfortable, not too hot and not too cold, you are more likely to do a good job at washing your hands. This was an example where we successfully argued our case before the Conference for Food Protection, which is a group that helps the FDA in what they put in the model food code, which gets adopted by different states to regulate grocery stores and supermarkets and supermarkets and restaurants. And the code originally required that a hand washing sink be capable of dispensing water at 100 degrees Fahrenheit. And thanks to our research and our efforts to change the code, that has now changed from requiring the sink to dispense water at 100 degrees to now to use language that's consistent with the plumbing code, which says that the hand wash sink has to be capable of dispensing tempered water. And tempered water is water, I think, that's in the temperature range of 70 to 90 degrees Fahrenheit. I forget exactly, but basically it makes it it's less likely for a food service operation to get dinged because their water temperature only goes to 95 or 99 degrees Fahrenheit. So again, water temperature doesn't matter, but really what matters is using a comfortable temperature. All right, so this is another example where we're trying to move policy in a direction that we think is supported by the science. And this is, again, some work that was funded by Gojo Corporation. They paid John Boyce and I for our time to write this work. They had no input into our conclusions, but they did fund our time. And for those of you, and in fact, you folks on the call, you probably know John Boyce more than you know me. John, in 2002, wrote a seminal document entitled Guideline for Hand Hygiene in Healthcare Settings, Recommendations of the Healthcare Infection Control Practices Advisory Committee, et cetera, and task force. And so what that document from 2002 did was it basically said, look, we should stop mandating hospitals to require hand washing because guess what? Use of alcohol-based hand sanitizers are just as effective and in fact would improve compliance. And so the work that John and his team did was really seminal in getting things changed in hospitals. And so what we did was we wrote a similar article focused on food service, and the title is Scientific Evidence Supports the Use of Alcohol-Based Hand Sanitizers as an Effective Alternative to Hand Washing in Retail Food and Food Service Settings when heavy soiling is not present on hands. And again, this was not original research. This was an analysis of existing research. And I'll dig into it a little bit. In one case, we summarize 4 other studies that looked at evaluating the effectiveness of food establishment hand washing. And what we found was that based on this analysis, that food workers, if they wanted to comply with the code, they would have to be washing their hands somewhere between 10 and 40 times an hour, which means that they would, by the time you figure the time to walk to a hand washing sink, the time to do a 20-second hand wash and then dry your hands and then walk back to whatever your next task was, these people would be spending a significant portion of every hour at the hand wash sink. And I don't know if you've ever tried washing your hands 40 times in an hour. The skin of your hands really doesn't like that. And you'll develop dermatitis, you'll develop cracking and red skin. It's really not a very nice thing to wash your hands that many times in an hour. And again, you can see that I guess the good news is, maybe it's not good news, but the bottom line is many people in full service or fast food restaurants are not washing their hands that many times an hour because they don't feel like their hands are dirty. And so our recommendation here is we would really like to see, in situations where your hands are not covered with poop or your hands are not covered with raw meat or raw meat juices, we'd really like to see the code allow people to use, much as a doctor can use in a hospital, right? Or a nurse can use an alcohol-based hand sanitizer as an alternative to a hand wash. So we have not been able to get any movement on that, but we're going to keep trying on that front as well. All right, so let me just give you sort of a summary in maybe no particular order except increasing number of words so that I could make a nice slanty word cloud there with my nice graphic there on the right. My advice to you, if you're asking for it, is you should avoid bulk refillable soap. Those things are nasty, and so I encourage you to not use those products. Or if you do use them, realize that about 15% of the time you may be actually adding bacteria onto your hands. Also, I think this is a message that might be somewhat controversial. Anything is better than nothing, right? Even if you do a splash and dash, that's going to get 90% of the bacteria off your hands. And so certainly if your hands are visibly soiled, or you have, again, poop or chicken juice or beef hamburger particles on your hands, that's the time when you want to do a good job with soap and water and 20 seconds. Using soap is better than not using soap. Water temperature doesn't matter. Use a water temperature that's comfortable for you. And again, I would encourage you, to think about using hand sanitizer as an alternative to hand washing. And certainly, we've seen this in the pandemic, right? Where I know if you guys are anything like me, you've got bottles of hand sanitizer around your house. So you're carrying one in your backpack or your purse. You probably have one in your car as well. Because guess what? If you don't have access to soap and water, hand sanitizer is really good. And the good news is it's especially good at killing wimpy viruses like SARS-CoV-2. Now, of course, the real risk of getting SARS-CoV-2 is probably not from getting it on your hands. The real risk is probably getting it from somebody who breathes or sneezes or coughs on you. But again, it's good to know we have hand sanitizers out there. Antibacterial soap is better than plain or bland soap, but unfortunately, because of changes mandated by the FDA Center for Drug Evaluation and Research, consumers can no longer buy antibacterial soaps containing triclosan. And so the only place that you'll see antibacterial soaps commonly is you might see them in doctors' offices, you might see them in restaurants and in food processing plants. There's still a possibility that FDA CDER is going to take those away from us, but we're working hard because we do think that antibacterial soaps do have a benefit. Remember that paper towels are great because they can help to remove bacteria from your hands. But another point that I didn't make that I'll make here is that one key way that bacteria transfer to and from a surface is when that surface is wet. And so if you leave the restroom with wet hands, you are much more likely to transfer bacteria from the door handle to your hands or from your hands to the door handle versus when your hands are dry. Dry surfaces don't transfer nearly as many bacteria as wet surfaces. Also keep in mind that hand washing is not magic. Hand washing does give you about a 99% reduction in the number of bacteria on your hands. That's about 2 orders of magnitude or 2 logs. But remember that that's not 100%, and that you may still, if you started off with a lot of bacteria on your hands, if you started off with 1 million or 100 million bacteria on your hands, after a good, perfect, well-executed hand wash, you're still going to have a lot of bacteria left on your hands. And in fact, when it comes to sick workers and people with bacteria on their hands, honestly, the best recommendation in terms of what to do with food is keep those people away from food. If you're sick with vomiting or diarrhea, I just don't want you anywhere near my food, whether or not you can do a good job washing your hands, because I know that there's a chance, a pretty significant chance, that you're still going to contaminate that food, even if you're washing your hands perfectly. Certainly, I think alcohol-based hand sanitizers as an alternative might work in food service. It certainly seems to have worked very well in hospitals, and so I would certainly like to see. We'll certainly like to see that come and be recognized in the food code. But again, probably not today, not tomorrow, but maybe at some point in the future. We'll keep working to try to develop food policy that's based on science. So with that, that is my last slide. I'd be happy to take any questions at this time.
Thank you, Dr. Schaffner. That was a really great presentation. Very informative. I do have a few questions here for you.
Sure.
All right. So why wouldn't adding new soap bring down the levels of bacteria that is growing? Are they immune?
Yeah, that's a really good question. So certainly there is some dilution that would happen if you were to add new soap to a bulk soap dispenser. What we think is happening, and I've not done this research, but others have done, and I'm familiar with it. What happens is you get a biofilm buildup, and so you basically get the soap dispenser colonized with bacteria. And then especially if you're dropping in soap that you're already diluting, such that, because there are nutrients in soap, but usually there's more things that are anti-nutritional. But especially if you're diluting that soap, there's really, you have like sort of weak soap to begin with, and then the bacteria see that and they start to eat and digest the product, and the levels go up. And again, they're in a biofilm, so they're kind of protected, and they'll keep sloughing cells off that biofilm into the soap dispenser.
Thank you. For bulk soap dispensers, were you testing the contents before it was placed in the dispensing unit?
No, we were not. We were randomly going around we tried for 300 locations. We ended up getting 297 locations. So about 100 locations each in Arizona, which was one investigator, New Jersey, which was me, and then Ohio, which was the folks from Gojo. And so we had graduate students or volunteers basically walking around to facilities and then secretly stealing some of their soap and bringing it back to the laboratory for testing. And so we didn't, and so the people whose soap we were sampling, unless they walked in on us in the bathroom, didn't know that we were testing their soaps.
Wow, interesting. Sanitizers are effective at reducing bacteria, but are they as helpful for humans as hand soap?
I think that in terms of the effectiveness, they're probably equivalent. I will say that both soaps and sanitizers do need to be properly formulated. And so again, I'll shill a little bit for Gojo. I do sit on their food safety advisory board. I am compensated for that. So they do have, there is a financial conflict of interest there. But I will say, and I would say this whether they gave me money or not, that they do make a very nice hand sanitizer, and you can use that repeatedly over time, and your hands will still feel very nice and very soft. If you use a cheap hand sanitizer or one that's not properly made, or if you just pour like rubbing alcohol on your hands, right, your hands will get chewed up by that, just like repeatedly washing your hands. And again, same thing for soap. Soaps can be properly formulated, so they'll be gentle on the hands. But even if you have a well-formulated soap, I think you're going to be hard pressed to use that soap 40 times in an hour and have your hands look okay. I'm not sure what using hand sanitizer 40 times in an hour will do, but certainly I know just from my own anecdotal experience, I find hand sanitizers to be gentler on my skin and gentler on my hands in general, especially if I'm using one that's a well-formulated one with, skin softeners and things like that to keep your skin healthy.
Great, thank you. How many times can we use hand sanitizers before it's unbeneficial?
I would say my advice is use your own judgment, right? It's not a matter of how many times you can use it before it's not beneficial. I would say pick the appropriate treatment, right? If I'm away from a sink with soap and I feel like my hands might become contaminated, I'll use hand sanitizer. If I'm working in the kitchen or I'm in the bathroom, right, and I've got poop on my hands, or I've been handling chicken or beef or fish or anything that might be contaminated, or if I've been working in the garden, right, or if I've been cleaning up after my dog and picking up their poop, those are the times when I'm going to want to use soap, right? When my hands are visibly physically soiled. Sanitizer is not really good at removing physical soil from the hands. Soap is really good at doing that. On the other hand, hand sanitizers are really effective at killing microorganisms when there's no debris on the hands, or even a small amount of debris.
Great, thank you. After you wash your hands, the sink will get all the bacteria. How will you be able to control the bacteria in the sink?
That's a really good question, and we don't often really think about that. But absolutely, you're washing those bacteria off of your hands, and they're going into the sink. Now, hopefully, most of the time they're going down the drain of the sink. But you're absolutely right; the sink can become contaminated. And in fact, surveys. I don't know of any surveys that have been done. I know in surveys in food processing plants, if you look at the drains, which is sort of like the sink of the food processing plant, the floor drains are often contaminated with bacteria. And I know when people have done looking for bacteria in households, the bathroom sink and the kitchen sink are often environments where you find microorganisms. And that's because in part we're washing them off our hands, but also what else do we find in sinks? Well, typically they're moist environments, and especially the sink in your kitchen, there may be food debris in that sink. And so bacteria need food and moisture. And if you let food and moisture accumulate in your sink, that's going to lead to increased concentrations of microorganisms. And so, yeah, realize that the sink is a focal point for cross-contamination. And I suppose the solution there is to clean and sanitize your sink on a regular basis.
Great, thank you. Do antimicrobial products like soap and sanitizers help create superbugs, or is the thinking different now?
No, and that is the logic, right? So the FDA Center for Drug Evaluation and Research laid out two statements when they banned triclosan for consumer soaps. They laid out one claim, which is to say these products don't work. There is no benefit. We've published work that is in contrast to that. So I believe these products do truly have a benefit. They also, in their logic, they also said, so number one, they don't have a benefit. And number two, they can accumulate in the environment and can have ecological consequences as a result of that. Now, I don't know how much of that is creation of superbugs so much as it is just ecological consequences from these toxins in the environment. And that's an area where I don't do research and I'm not really qualified to comment. But again, like I said, I was disappointed in that they appeared to discount the effectiveness of these products because I do think that they are effective. But again, that's the game, right? Like sometimes a regulatory agency will do something that you don't agree with. Sometimes they'll do something you do agree with, and so that's just the game.
Great, thank you. Is there a difference you found in the research between the push dispensers and the non-touch ones? Bacteria found?
That's a good question. We did not specifically look at this. Like I said, the work that we did at the beginning with Sloan Valve was on touch-free faucets, not touch-free soap dispensers. But obviously, if you have a touch-free faucet, and I'm assuming if you have a touch-free soap dispenser, that dispenser is never going to become contaminated with bacteria from your hands. And we were thinking about the faucet handles specifically because when you go to wash your hands, you've got dirty hands. So you touch the faucet handles and you contaminate them. You wash your hands, you remove bacteria from your hands, and then you go back to turn the faucet off by, again, touching with your hands. And so the idea was, well, what kind of transfer would we get? And what we see through computer simulation is basically there's a sort of a baseline level of contamination that stays on those faucet handles, kind of jumping back and forth between hands and handles and hands and handles. And so you will, at least our simulations would predict that you just continually have that level of contamination. And having a touch-free system takes that risk out of the system.
Great, thank you. Wondering if bacteria hang out and get kicked up from the surface in the air dryer back into the hands.
Yeah, it's a good question. We've not done research on hot air hand dryers, but there is a well-established literature on the topic. And we do know that these hot air hand dryers, at least some models, will spread bacteria, either blow the bacteria back onto you or blow them into the environment, and then they can spread from there. I think there's been some examples where hot air hand dryers have become colonized with bacteria, and so again, I think these are. I understand the logic why somebody would want to use them to get rid of paper towel waste, but there's no risk-free ride here, right? There's always going to be risk trade-offs with everything you choose.
Okay, great. Should hand sanitizers be used after expiration date?
That's a really good question. And I'd say probably that here's the thing. If you have no other hand sanitizer to use, I would go ahead and use the expired one versus not using it. Okay. But realize that these products do have an expiration date. What can happen is over time, the ethanol in them can evaporate. And if it's evaporated, it's not in the product anymore. So the product is not going to be as effective. And so, and then also I've had some that it's been sitting around my office for years, and you can tell that the color, even high quality Purell, right? The color degrades, they get tacky and sticky, again, partly because they've lost moisture and they've lost alcohol. So again, better than nothing, but given a choice, you should use stuff that's not expired.
Okay, which goes to my next question. Does keeping hand sanitizer in the car, which I'm guilty of, does the heat decrease its effectiveness?
Yeah, it's not that the heat will necessarily decrease its effectiveness, but if it's in a hot car and it's not sealed, again, that ethanol can evaporate. And so as a best practice, well, here's what I would say. If you're going to keep it in your car, keep a small bottle in your car. We have in one of our cars, do as I say, not as I do. In one of our cars, we have a giant bottle that we've had from early in the pandemic, and it's been in that hot car a long time. It still feels like it. You still feel the cooling effect. It still feels like it has ethanol in it. But again, my advice is keep a small bottle in your car, and then when it runs out, put in a fresh small bottle. That's going to ensure that you don't have a big bottle that's just completely devoid of any active agent.
Okay, and I have two more questions. Do alcohol-based hand sanitizers kill norovirus?
Excellent question. I don't know if I have time in the time we have remaining to answer it, but I'll do my best. The short answer is it's complicated and it depends. I'm not wearing my "It's complicated and it depends" t-shirt today. Otherwise, I'd show it to you. But yeah, so there are ways of formulating alcohol-based. So even conventional alcohol-based hand sanitizer does seem to have some effectiveness against norovirus, but as we know, norovirus is a non-envelope virus. It's not a wimpy virus like SARS-CoV-2. It's a pretty hardy virus. The good news is that you can formulate alcohol-based hand sanitizers with extra ingredients that do make them more effective against norovirus. And again, the Gojo company, makers of Purell, again, I have a consulting relationship with them, but they publish papers, I've not, but these are papers that they've published with other reputable scientists, which show that, yes, there are ways you can formulate hand sanitizers to be effective against norovirus. Where you run into trouble is because of the way these products are regulated, you can't actually make claims on the label. If you make claims on the label, that you get into trouble with the regulators. But there's plenty of examples in the scientific literature of products that are what they call the VF, the virus formulation, that are effective against norovirus. But they can't actually tell you that in the marketing materials, otherwise they get in trouble for violating the law. Again, and that's more a reflection of the fact that I think our laws in this case are kind of archaic and have not advanced with the way the science has advanced.
Okay, great. I know we have a few other questions, but I have to be respectful of everyone's time. Dr. Schaffner, if anything, if I could send those questions over to you.
Sure.
But
please do.
Those who are asking about credits, there is an evaluation at the end. Please check off the credits that you're seeking, and we'll make sure we get that over to you. Dr. Schaffner, thank you so much for this wonderful presentation. As you can see, we had a lot of questions. And thank you everyone for joining today's webinar. Please look out for our next webinar that will be held in July. And thank you again, Dr. Schaffner.
Sure. Thanks to you, Kendra, and to the Center for inviting me. I really appreciate it.
You're most welcome. Thank you. Take care, everyone.